Okay, so this is a video that's going to guide you through some of the problems for our human genetics lab. So the first activity, we're not going to be in class. So the PTC is a compound that some individuals can taste and some can't. So if we were in class, you would all be given a piece of paper and you would say, can you taste the paper or not? And you would write that down. Are you a taster or a non-taster? So you can just write down, you can leave that blank actually. For me, I know that I'm a taster, so I put taster here. And then I would look at the class results. So I just did this in my class and we found out that we actually had, I think we had like 8 people that were tasters and 9 people that were non-tasters. And it was very odd because generally most of the population is tasting. So are the results expected? No. And why would that be? Well, because we have a very small sample size. Small sample size. Okay. So with the small sample size, you're not getting a good estimate of the true population value. So how is that green? Because it's all right. If I should use red. Is that better? I'm trying to, yeah, I think the red's better. Okay, so I'm going to switch to red. So tasters, tasting is a dominant allele, which is represented with a capital. So the genotype for tasters is either going to be homozygous dominant, which is 2 capital T's, or heterozygous, which is a capital T and a lowercase t. Non-taster is a recessive phenotype. The physical expression of the genotype is 2 recessive alleles or 2 lowercase t's. All right, so that sets us up for actually what you can do for this activity. So in this activity, we have a pedigree chart, and this shows inheritance patterns. So you can see the phenotype. It's represented by a legend, which tells you if an individual is a PTC taster or a non-taster. So in this case, the non-tasters are open, so they're not shaded, and the tasters are shaded. So you're given instructions that for this pedigree chart, you need to determine the genotypes of all the people. So you're going to write it right above the circle or the square. So for John, he is a non-taster, and so non-tasters we know are recessive. It's a recessive phenotype. So all of the open boxes, you can go ahead and put 2 lowercase t's or homozygous recessive genotype for all of the non-tasters. So in this case, you can just fill those in. What's harder is if you are a taster, which is indicated by this black shaded circle or square. A circle is female and the square is male. Okay. So Joan, she could either be, if we look back, she could either be homozygous dominant or heterozygous and be a taster. So we really have to look at the offspring and work our way back to figure out her genotype. So because she has offspring that are non-tasters, we know she has to be able to give them a recessive allele. So we know she has to be heterozygous. So you can do that for all of them. In some cases, you may not be able to determine if it's heterozygous or homozygous dominant. So like here with Elizabeth, both of her parents are tasters. She's a taster. Her offspring, she has a non-taster as an offspring. So we know, again, that's homozygous recessive. So that means Elizabeth has to be heterozygous because she has to be able to give her offspring a recessive allele. One allele comes from both parents. And we know Tom, even though he has 2, he can only give one of his alleles to the offspring. So we know that Elizabeth is heterozygous, but we still don't know if her parents are heterozygous or homozygous. So we have to put T capital T question mark, capital T question mark, like that. Okay. All right, and we know one of them would have to be heterozygous, but we don't know which one, and so this is how you would indicate it. So you're going to go through and complete all of the rest of these, and then answer this question here, what's by making a Punnett square. So you refer to figure 1. If Jill marries Howard, so you go and you figure out Jill's genotype, you know that she's homozygous recessive. Howard, well, we'd have to figure out what his genotype is. So since his mother, Rita, is homozygous recessive, she's going to give Howard one of those alleles because that's all she can give. So we know Howard is heterozygous. So, okay. So then you do a Punnett square. So then you basically set up your Punnett square. In this class, I prefer if your males go on the top and your females go on the side, just as convention. So Howard is going to give either a dominant allele or recessive allele. Jill is going to give 2 recessive alleles. You complete the Punnett square, and then you figure out the probability that their children are tasters or non-tasters. So in here, you're going to put a number between 0 and 1. Okay. All right. Okay. And I think that's all you need for activity 1. You'll be answering the questions in the comprehension check, so make sure you do that. Okay. Activity 2, you're going to be looking at sex-linked traits. So these are inherited, as an inheritance pattern that is passed down through the sex chromosome, so on X or Y. If it's X-linked, then the trait is actually on the X chromosome. And so there will be a different expression of X-linked traits in males and females. Females have 2 X chromosomes, males have an X and a Y. So basically, if you're female, to have a recessive phenotype, you have to get 2 recessive alleles, and if you're male, you only need 1 because the recessive, because there's nothing on the Y. So it's much easier for males to express recessive traits. Okay, so then you'll go down. What I just want to point out is please use this notation. So, in this, I said males and females, but then I noticed there's a different example here. So we're going to go with this, the females on the top in this one. Anytime you have a female, you're going to have X's. Anytime, just 2 X's. Anytime you have a male, you have X and a Y. So what I could do is I could actually go in and fill out the X's and the Y's right away as my first step when I'm doing sex-linked traits. And then I go back and look at the alleles, which are the superscripts here. So then I put the alleles in. Okay. All right. And I'll let you read through the background on this for more of an understanding of hemophilia. And you can answer the questions. So in this case, we have a 0 indicated here for Y. That just means that the Y has nothing on it. It's not actually an allele. So this case, if it's an X and a Y, X or Y, so these 2 are males, right? So these are male offspring because they have X and Y. These 2 boxes represent female offspring because they're 2 X's. Here we've got a small h comes down. So I'll just give you this one. So this one, H is small. The small represents a recessive trait, and hemophilia is recessive. So in this case, this male has hemophilia. Okay, this male has hemophilia. All right. Okay, so you can finish that. You also have a pedigree chart associated with hemophilia. So in this case, when you're creating your pedigree, you're going to write out your genotypes. So you're always going to have the males are going to be XY, the females are going to be XX. So you could go ahead and write all that out for everyone first. Please make it clear. Okay, and you'll write them out. This one we know for sure. Male has hemophilia. Hemophilia is a recessive trait, so we know this is a lowercase h. And it's only found on the X chromosome, and so it's only on the X. Leopold had to get this recessive trait from one of his parents. Okay. If the dad had it, he would express it because he only has one place to have it. So the dad doesn't have it. So that means normal blood clotting, which is capital H. The mom, Victoria, also does not express hemophilia. So she has to have a normal allele, but we know that she passed on a recessive allele. So we can work backwards. So you can fill out the rest of it using that methodology. Okay. All right. Okay, please make sure you do the challenge questions. Okay. And next. Okay, so next we have sickle cell anemia. This is codominance. So in sickle cell anemia, we have a sickle trait, which is codominant with normal blood, red blood cell shape. So the sickle shape is expressed and the normal shape is expressed. So how the notation looks for this is we are using 2 letters as the gene for the, basically for this shape of the blood cell, which is a result of hemoglobin. So it's called HB. And you can, if you have 2 of the alleles A, then it's normal blood. You have normal shaped red blood cells. If you have A and S, you have a sickle cell trait. That means half of your blood cells are going to be round and half are going to be sickle shaped. Whereas if you're normal, all of your blood is going to be normal. If you're sickle cell anemia, all of your blood is going to be sickle shaped. Now, it's only going to be sickle shaped in environments with low oxygen. That's what triggers a change in the conformation of the proteins, hemoglobin. But all of the cells, if you have sickle cell anemia, can take on this shape in the correct environment. So this is sickle cell anemia. If you have 1 copy of the mutated allele, it's sickle cell trait. And if you have 2 normal alleles, it's normal. So when you're writing, again, you're going to put HB in all of these. Okay. I know it takes a minute to do these letters, but please go through with that. And then you can worry about your superscripts. Okay. So I did all that. Now I can worry about my superscripts. If both parents are heterozygous, that means they have 1 of each allele. What are the possible genotypes and phenotypes? So that means they're heterozygous, so I'm going to put an A and an S, and an A and an S. And then they're going to be inherited the same way you would do a normal Punnett square, right? A, S, 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 okay? So then over here, like you can see the genotypes, right? But the phenotype question you have to answer. So the phenotypes, these are the phenotypes up here. So it's either normal, sickle cell trait, or sickle cell anemia, okay? So you basically want to give the probability of each of the genotypes. So I'll go ahead and do this one. So, so for the phenotypes, the normal, trait, and the sickle cell anemia. So normal would be 2 A's. So we've got 0.25 because it's 1 out of 4. The trait is one of each allele, so that's 0.5. And then anemia is going to be 0.25, 1 out of 4. All right, so you can do this one. Probability is a number between 0 and 1. Okay, so you can do the Punnett square, but then also state the probability. You won't get full credit unless you actually answer the question and show your work. Okay, so show work and answer the question. All right. Okay, so now we're to activity 4. So activity 4 is we're looking at pedigree charts with sickle cell disease as our context. And we're looking at in the context of malaria. So malaria is a disease that is caused by protists called Plasmodium, transferred by mosquitoes. And it turns out that there is a relationship between the severity of malaria and the mortality of malaria and sickle cell disease. And that's what you're going to be investigating. What is that relationship? So you'll read through this. In this pedigree chart, it's now showing these half shaded cells, and those half shaded cells are carriers. So that's different than before. The previous pedigree chart did not show half shading for carriers. Okay, so there we go. You are going to, in this one, just follow the directions. So you're going to read through it. These are numbered here, so you can see 1 through 38. And then here you can see the ones that currently have malaria. It refers to this chart. So you'll answer these questions. And it says, on the pedigree chart, place the letter M next to the numbers of all persons who have malaria or who have died from malaria. So, malaria 13. So you're going to put an M here, right? M. And then 32. M. And then 33. M. Okay. Then the next one, H. The healthy ones, right? So that's basically what you're going to do. So just make sure you're going through and following the directions, and then you're going to answer this question. What is the most common genotype for people who have or have died from malaria? Okay, so you're going to go back and look at the genotype. These are the genotypes. So it's going to be one of these 3 answers that you put in this open blank. Healthy individuals, it's going to be one of these 3 answers that you put in this blank. Okay. Okay. So for this one, you're just figuring out the percent mortality. So the total, you're calculating percents. So this one, the number that's dead is 8, a total of 15. So 8 divided by 15 times 100, put that number in here, right? So you do that for each of these, it should add up to 100%. You do the same thing over here with neighboring communities. You're basically looking at the percentage, which you calculate by taking the number of the interested quantity divided by the total times 100, and you get your percent, okay? Again, it should add up to 100. Answer these questions. Answer this questions. Right, show your work. So make sure you, for these, again, you're making the Punnett square. You're using the correct notation. So the notation is shown here. Okay, right? HB superscript A, HB superscript A. So please take the time to use that correct notation. And then you're answering the question about the probability. If you only do half of that, you're only going to get half the points, okay? So just, I want you to all be successful, so please make sure you're paying attention to that. You can answer these questions. Yeah, okay. Again, answer the challenge questions. And then the last activity is on codominance and multiple alleles. It's on blood type. So there's some different notations out there for blood type. We're going to use this one for our activity to be consistent. So in human blood types, the ABO blood type, we're going to use the letter I for that, the gene that codes for ABO blood type. And then there's going to be 3 different alleles. There's A, and then there's B, and then there's O. Oh, they're using it different. Okay, so shoot, that's kind of annoying. So then anyway, we're going to stick with it. So we're going to use this recessive allele here, I. So the idea here is that A and B are codominant, so they're both expressed when they're present. So one does not mask the other, and one is not dominant to the other. They're codominant. I is the recessive allele, and that means it's masked by B or A. Okay, so I is going to correspond to, let's look over here. Sorry, I'm just going to go to this table. So this table shows you the relationship between phenotypes, the expressed ABO blood type, and the possible genotypes. So for O, the only way to get type O blood is to have 2 recessive alleles, 2 little i's. The only way to get type AB blood is to have 1 A allele and 1 B allele. But now there's 2 genotypes for type B and 2 genotypes for type A. So for B, you could have either homozygous for B, or you could have 1 B and 1 O. Remember, B is dominant to O, so it's type B. If you just have 1 dominant allele, it's going to be expressed. Same thing for A. So for A, there's 2 possible genotypes. You can have 2 A alleles, or you can have an A and an O allele. All right, so for this last part here, you are going to explain your answer. You can use a Punnett square. I would suggest using a Punnett square to answer this, unless you can really articulate your answer well in words. If so, I will give you credit, but I find that it's just easier to show with a Punnett square. And then as well, down here, if you can just show a cross where it actually works out, show the offspring as 0 or type O, then you're good, and you don't have to use all kinds of words to explain it. Okay, and then here's the last part, the self-test. So the self-test is optional. You can always do that if you want to, but you are not required to do the self-test. So activities 1 through 5, that's your guidance. You can email me if you have questions, and I hope that was helpful.